Welcome to the study of hematology. Hematology is the study of blood. So within this chapter, we're going to look at all aspects of blood, and we're certainly going to be introduced to the basic components and what those components' functions are, and then even look at some pathology associated with blood. So when we think about the cardiovascular system, we understand that this actually includes not just the pump, the heart, or the vessels where blood is being sent to, but also blood itself. So this is what we're going to start with in our cardiovascular system section. We're going to start with hematology, the study of blood, and then the next chapter deals with the heart, and then the next chapter after that deals with the vessels. So when we think about the functions of this, obviously, most of our cells are stationary. So you have a kidney that has cells that are very specific in their functions to the kidney. Your liver, same thing. Lungs, same thing. But for those cells, they have to get nutrients. So how nutrients are delivered, and nutrients in the form of gases like oxygen and the removal of CO2, waste products, hormones, and whatnot, how they're delivered is through the bloodstream. So blood is actually a liquid tissue that is part of the connective tissues that we studied when we were looking at histology. And some of those functions are to supply nutrients to the tissues and remove waste, but also protect, so delivering the cells that are needed, that are part of the white blood cells that protect us, destroying microorganisms, and cancer. Cancer cells are happening all the time, but they're being recognized by our immune system cells and they're being destroyed. So fluid balance is something that I think also students tend to not really appreciate until they've gotten into the study of hematology. But how much volume, fluid volume, your blood actually has is going to be directly related to your blood pressure. So the higher your blood volume, the higher the pressure being exerted on the vascular walls of those blood vessels. So controlling blood pressure, fluid balance, stabilizing pH, all of these functions are functions of blood. Temperature regulation. So when you're really hot, warm, you've got to remove, that blood ends up going to the peripheral structures. Your vessels get larger in your hands and arms and legs, and it disperses heat. When you're cold, those vessels will contract down. Blood will be directed toward your visceral organs, your internal organs, to maintain the temperature that you need and not for it to get too cold. So all of these are actually functions of the circulatory system. And when we think about blood, depending on the size of the person, slight interruption there, sorry about that. But when we think about blood, it is a liquid tissue, a connective tissue, and depending on the size of the person, adults, larger adults have more blood than smaller adults, but roughly 4 to 6 liters are circulating around within the vascular system, and obviously it's the heart, the pump, that is pumping that blood around. So when we think about that it's a liquid tissue, if we were to spin blood down, take blood into a vial and spin it down using centrifugal force, the cells would go to the bottom of the tube, and what would be at the top would be the liquid part, the extracellular substance, the extracellular matrix, which in blood is called plasma. Plasma should be clear with a slight, just maybe a slight yellow tinge, but it should be clear because it's predominantly water. So the major part of whole blood is actually plasma, more than 50% usually, and most of that is water. The cellular components, the formed elements, or the cellular components, include red blood cells, which are called erythrocytes, white blood cells, which are known as leukocytes, and then platelets, which are called thrombocytes. You'll see those terms in just a minute. If we were to take whole blood and take a drop and make a blood film on a microscope slide and take a look at that blood, we stain it with a right stain, which is a stain that gets picked up by the cells, and especially the nuclei of white blood cells. But we would see under a microscope, using about 400 magnification, we would see these cells that look anucleated, meaning they are without a nucleus. These cells are the red blood cells, and you can see that by far and away they outnumber the nucleated cells, which are the white blood cells. So the predominant cellular component are erythrocytes, red blood cells, and then you are going to have white blood cells that are nucleated, and then these partial little cells that almost look like they're pieces of a cell that are called platelets, or more accurately referred to as thrombocytes. So again, when we're thinking about the components of blood and what makes up blood, we do know that there are these different formed elements. Leukocytes, there are many different kinds of white blood cells. White blood cells aren't all created equal. They look different from each other. Neutrophils look different from eosinophils versus basophils, lymphocytes, monocytes. They look different from each other because they have very different functions. When we think about red blood cells, we know that the major function is to deliver oxygen to our cells that make up our tissues, and to pick up CO2 and bring it back to the lungs so that we can breathe CO2 out as a waste product. Platelets, when we think about platelets or thrombocytes, these are going to function in blood clotting. But white blood cells protect us. So if we had to have one word, we would say immunity, or maybe that word protection. To protect, that's their function. But they're not all created equal. Neutrophils protect against bacteria. Eosinophils actually are going to respond to allergies and parasites. Basophils can also respond to allergens, but also to other things like viruses as well. Lymphocytes are going to respond to viruses. We typically think about viral infections, lymphocyte counts going up, but also in cancer to help to fight against cancerous cells. And monocytes act as our big eaters. We call them big eaters, macrophages. And they are going to act like the dump trucks of the system. They're going to go around gobbling up debris. So cells and tissues that have been damaged or destroyed, but also some foreign particles. So monocytes act as those macrophages, big eaters. So again, we have different types of white blood cells. Ones that contain granules that I've just mentioned, and ones that don't have any granules. But again, all of them need to be present in the amounts that we expect them to be, so their percentages of the total need to be just right, and they have very specific functions. The reason that it's important to understand the percentages that should be there of the total white count, so there's a total white blood cell count, but then that's broken down into percentages of these cells, is because when you have a bacterial infection, these are the cells that are going to be increased. And that can tell you, it can diagnose that for you. So just getting a white count and then breaking it down to how many neutrophils are present, if that count is higher than you expected, then you know you're dealing most likely with a bacterial infection. If the eosinophil count is high, then it's either allergies or worms, parasites. Lymphocytes, we're thinking if that count is higher than expected, that percentage of the total white is higher, we're thinking viruses typically. And monocytes will tend to increase no matter what you're fighting, no matter whether it's bacterial or viral or even allergic, the white counts go up. This is an illustration, so drawing, it's a very good drawing, of what these nucleated white cells look like and how they look different from each other. But structure will always dictate function, so anatomy always goes hand in hand with function. These monocytes are the ones that aggregate together to become macrophages. Eosinophils have these huge orangey pink granules when you stain them with the right stain. These granules that are included inside the cells contain histamines. Histamines are what we know go up when someone's having an allergic. Histamines have a very important function, so they get a bad name. But actually, some of the things that they can do are initiate pain receptors, but also cause vessels to dilate, relax. and can also cause capillaries to be a little bit more leaky, meaning that water will diffuse into the tissue spaces more readily if histamines are present. But these granules contain histamines. Neutrophils have little tiny granules that have antimicrobial effects that fight against bacteria. And then lymphocytes, we're going to find out when we get to the lymphatic system chapter that there are several different kinds of lymphocytes. But for right now, let's just use lymphocyte as an umbrella term and understand that lymphocytes do a fabulous job at actually protecting us against viral infections and also fighting against cancer cells that have lost their control mechanisms, life cycle control mechanisms, and actually can lead to cancerous changes. These are basophils, which it's hard in this illustration to see the nuclei, but the nuclei are often bilobed, just like this eosinophil one. The granules are so huge and take up the stain so well. These granules contain heparin and also histamine, but mostly these are staining this way because of the heparin. Heparin is an anticoagulant, which means, if I break that word down for you, it means it helps you to prevent unwanted blood clotting. Blood clotting is wonderful. We need it all the time. We're always doing things that break little capillaries, but you have to have blood clotting to stop from bleeding, and you have the mechanisms in place for that normally, but you don't want unwanted blood clotting. You don't want too much blood clotting, and these basophils help with that by containing heparin. So if we were to take, again, a tube of blood, and probably most of you have had a phlebotomy procedure where blood is taken from a vein, and in the tube we were to spin it down, we would know that the cells would be packed in the bottom of that tube because of centrifugal force, and we would see that the predominant, roughly over 50% for most people, is really plasma, which again is predominantly water. Now, when we think about it being predominantly water, we know that it also has other things in it that are very good. It has hormones that we just finished talking about, hormones, waste products, even some gases that are dissolved. So all kinds of nutrients, glucose is going to be found, and we know we need that for energy, for that aerobic respiration. So all kinds of electrolytes, sodium and many other electrolytes as well. So this is in plasma, but predominantly it's water. And the packed cells, we said, are mostly erythrocytes. There will be a little thin layer called a buffy coat that contains the white cells and the platelets. They are really a very small total percent of whole blood. When someone donates blood and gives a unit of blood at a blood bank, their blood is not kept whole. They take that unit of blood and they spin it down using centrifugal force, and they take the red cells, and those are referred to as packed cells. So when someone gets a blood transfusion, they're not getting whole blood from a donor. They're getting the packed cells. And they have to be given, these packed cells have to be kept in a certain temperature and in a certain environment, and they must be given usually within 60 to 90 days. The reason being is that red blood cells only survive about 120 days, and then they die. They have to be recycled. So they survive in the circulating peripheral blood for about 120 days. So packed cells must be given in a shorter time period than that, obviously, so that you make sure that the ones that are being given, enough of them will actually be functioning for a short period of time, and the person that was given the packed cells. The plasma is actually taken off and put into a new little plastic unit and frozen. So maybe you've heard of fresh frozen plasma. Plasma can be saved up to 1 year. But after a year, that would be sort of the expiration date as well on plasma, and it would have to be thrown out. The beauty about our blood banking system, whether it's the Red Cross or the American Heart Association systems, they are so good at using every bit of blood products and not wasting any. So they have a very good logistical center as far as like making sure that the blood that's donated is, almost all of it is actually used. The buffy coat that contains white cells and platelets, that's also removed separately. And it's irradiated because white blood cells cannot be donated. Someone's white blood cells can't be given to someone else because they would think that someone else is foreign and they would fight against that blood. So this buffy coat gets irradiated, so the white blood cells are destroyed, and the platelets are saved. And the platelets can be donated to someone else. So if you think about it, when you give a unit of blood, potentially there are 3 lives you could save. The packed cells could go to a donor, a patient. The platelets could go to another patient to save that patient, and a completely different patient would get the plasma that could be life-saving for them. So donating a unit of blood is really, a wonderful thing to do if you're able to do that. When we think about blood plasma, let's look a little bit deeper into that. So we know it's mostly water, but there are a lot of other substances that we talked about too, and you do want to realize that it is a complex mixture of water and then very important other substances. But when we think about it, you've sometimes heard about serum. Serum is essentially blood plasma without blood clotting factors. So it's without the blood clotting factors. A factor is a protein, and proteins are molecules. And if you were to take those blood clotting proteins out, now it's called serum. So that's the difference. If it's sort of technical, but sometimes certain tests require not plasma, it requires serum. So that's what you would need to do. You would need to make sure that you're putting it in a tube where the blood clots. If the blood's allowed to clot, the clot is going to pull all of the blood clotting factors into the clot. And now what's left when you spin it down is actually serum. So you probably all, when you've had your blood drawn, the different color top tubes have different preservatives in them. There's a couple that have nothing in them. And if there's nothing in them to keep them from clotting, blood will clot. When it slows down, when it stops circulating and moving, it will clot. So those tubes allow the blood to clot. And then when it's spun down, what's left over at the top is actually not plasma. It's serum, if that's happened. Now, the major plasma proteins, and you should know this, albumin. Albumins are a major protein. They are provided by the liver. The liver produces not only our blood clotting factors, but also albumins. So if someone has a problem with their liver, then one of the first clues we get is that they have bleeding problems. They may be bruising much more easily than they should be, but also the albumin levels are low if the liver is not functioning, and this contributes to blood thickness. Blood thickness being just right in homeostasis is really important. The amount of cells that you have, the percentage of erythrocytes, in this example is 45%, the amount of cells you have combined with albumin contribute to the thickness of blood. And blood cannot be too thin, and it should not be too thick. And the way that I can explain this to you, because this is important to understand that it influences blood pressure, fluid balance, and everything else, is I want you to think about your heart as a muscle, cardiac muscle, right? And it's pumping blood through miles and miles of vessels. So this pump is having to pump blood that goes to your fingertips, your toes, your hair follicles. It has a long way to go. The pump is having to pump it. If blood is too thick, it's hard on the heart to pump it. But if blood is too thin, it's also very hard on the heart. And it changes the osmolarity of the blood so that water would be moving. If it's too thin, it would be moving from the blood into the tissue spaces. If you remember the concept of osmosis, water moves down its concentration gradient, and there's really nothing your cells can do about that. For diffusion and movement of solutes, your cells can move solutes against their concentration gradients from low to high. But your cells can't move water from low to high. So the thickness, the osmolarity of blood has to be just right, because if it's not, water will end up moving down its concentration gradients, which can lead to edema. Edema, E-D-E-M-A. Edema means an excess of fluid in the tissue spaces. Now, if that's happening in the ankles and in the fingers, then that's, one thing. And usually just moving can help you disperse those fluids and get rid of the edema in those places. But if it is happening in the lungs, that's called pulmonary edema. It means the lungs can't fill with air and can't get rid of air the way they should. So air is impeded from moving. Pulmonary edema is not a good thing. If there's edema around the heart, then the heart can't fill with blood and pump it effectively either. It's being compressed. You can even have cerebral edema, which means that there's fluid pressing on your brain and central nervous system, which you can imagine is a life-threatening event. So thickness and osmolarity of blood is vital to be maintained within homeostasis, and what contributes the most to it are albumin, these proteins that are from the liver, and also the amount of red blood cells. I want you to know a term called hematocrit. Hematocrit is actually the percentage, and I know it's kind of weird the way it's written here, but the hematocrit value for this example would be 45%, just this example that they gave you. That would be the hematocrit number, 45%. The hematocrit is actually the percentage of whole blood that is cellular. And really, the predominant cells are erythrocytes. So the buffy coat really doesn't even contribute to that. It's less than 1% usually. So the percentage of whole blood that is cellular is the hematocrit value. It's very important when you're getting a blood test in hematology. They're called complete blood counts, a CBC. Complete blood counts include hematocrit. They include a total erythrocyte number per microliter of blood. The hematocrit's in percentage. The erythrocyte count is in total number in millions per microliter. A total white count with a breakdown of all the individual kinds of white cells. A CBC has a lot of test results. A thrombocyte count, and it's also going to give you, a couple of other sets of information as well. So a CBC is a really valuable tool in diagnostic medicine, and someone can have a CBC result back within minutes of having their blood drawn. So it is a valuable diagnostic tool to get a complete blood count, which includes all of these values. So again, some other plasma proteins that you find are antibodies. When you hear that someone has made antibodies to a certain foreign antigen, it means that they've built a little molecule, a protein molecule, that protects them. So antibodies we think of as being incredibly protective, right? And you're hearing a lot about it with immunizations now. So immunizations provide you a way to make antibodies without ever having to get sick, which is a beautiful thing. With the advent of immunizations, the world has changed as we knew it, as little as 60 years ago. The world is a different place because of immunizations. 60, 80 years ago, it was a 50-50 chance your child would make it to 5 years old. There were some places cultures that didn't even name a child until after they were 2 years old, because so many childhood illnesses and so many diseases made it unlikely that they would make it to that age, and so they wouldn't name the child. But anyway, it was a very different time, even in my grandmother's time and even my mother's time, where polio and smallpox were real things. Measles, mumps, rubella, pertussis, all of those, diphtheria, all of those, and now COVID, all of these things that had high mortality rates. So now we can have immunizations, we can get antibodies made, and they are incredibly protective. Fibrinogen is going to be one of those clotting factors. It's one of many that is circulating around in case you need to have a blood clot form. Like I said, you stub your toe, or you floss too hard, or you sneeze too hard, or whatever you might do, that you need to have blood clots formed. And blood clots sometimes are actually formed in the tissue spaces, and we call that loss of blood into the tissue spaces, with blood clots forming a bruise, don't we, a hematoma. But anyway, that's normal and it's natural and it happens daily. Most of the time we don't even know what's happening because it's at a small level. It stops so quickly. But fibrinogen is one of those many clotting factors that the liver again contributes to. So there are also other types of compounds. We have amino acids that we know are the monomers that end up making up our proteins. There are 20 different amino acids circulating around so that when our cells need to build a protein, which is just a chain of amino acids, it can do them. It has the building materials to do that. We also have breakdown products of protein that happen because proteins aren't made and last forever. Proteins have a shelf life, and as they're breaking down and products are being recycled, like many of the amino acids get recycled as proteins break down, there are also sometimes nitrogenous waste form that will end up being converted to urea. And urea can be gotten rid of through the kidneys. So as the kidneys are filtering blood plasma, because that's what the kidneys do, filter blood plasma, urine is simply filtered blood plasma, then one of the components that we call waste products are urea, and we'll talk more about that when we get to the excretory system, the urinary system. There are also incredible nutrients in blood plasma. I had mentioned glucose, but there's vitamins, fats, all kinds of things that the cells need as building materials to make the cells actually function. There's dissolved gases. There are electrolytes, which we know are ions, which are supplying the electricity, allowing for action potentials and resting potentials to be maintained. So we understand that sodium is the predominant extracellular blood plasma, extracellular cation, which is the positive ion. Positive ions. They're also negative ions. Okay, so blood viscosity and osmolarity I've already talked to you about a little bit, but again, this is just the idea of how resistant is this to flow. You can think about a pitcher, if a pitcher on a baseball mound, most of us have been to a baseball game. So if a pitcher has a baseball, and they are on the mound because they can pitch, obviously, he can get that baseball, the weight and the resistance of that baseball pretty easily over the plate. Rarely does it ever bounce before it gets to the plate. He can get it to the mitt, right? But if you gave that pitcher something heavier, if you gave it gave that pitcher, it's used to a baseball, but maybe gave him a softball or even a bocce ball, those are heavy, and ask him to throw the same, it's too heavy, it's too thick, and it would be really hard on the pitcher's arm to do that. So too thick is bad. The blood being too thick would be bad. But if it's too thin, if you gave that pitcher and told him he had to stay on the mound and you gave him like a wiffle ball or a ping pong ball that's light, too light, then that's hard too. That's going to be hard on the muscle as well. Both of those things would be very difficult for the pump and would make the pump work harder. But not only that, it also changes the osmolarity. And we said water moves down its grade. So again, thinking about viscosity is important. The percentage of those proteins in the plasma, but like albumins, but also the amount, the hematocrit. The percentage of the whole blood that is red blood cells, that is cellular. That's important too, because that makes us understand the viscosity of the blood. So again, I'll kind of go. So if somebody is hypoproteinemic, which means that hypo means low, protein obviously means this organic molecule we call protein made of monomers, amino acids. Emia as a suffix always means in the bloodstream. that always means that in the bloodstream. So low protein in the bloodstream. This can happen because of starvation or just poor diets, malnutrition. It can happen because of liver and or kidney disease as well. Burns, the skin is a major player in also keeping these in. So what can happen is that this changes the osmolarity, and what you'll see are like people with wasting disease, so like their muscles are wasting away, atrophying away, being eaten up really, but really large bellies. So ascites can actually be, which is the fluid building up in the abdominal cavity. You can kind of see that. Swollen abdomens, but at the same time very thin and emaciated looking arms and legs. Kwashiorkor is what that term is for children with severe malnutrition. And again, this is a life-threatening, at this time it's becoming a life-threatening event because of what it's doing to the cardiovascular system as well as multiple other organ systems. So when we think about how blood is produced, that's going to be the next topic that I talk about in the next lecture. So I'll stop this one right now, and we will have hematology probably in 3 parts. See you next time.